Hello everyone, and thank you for joining this presentation. Today, we are pleased to present part of the doctoral research of Veronica Rodriguez Celestino, a PhD student at the University of Guadalajara, in Mexico. Her research focuses on the design of hydroxylated C60-PORPHYRIN derivatives for the dual modulation of PTP1B and oxidative stress. This project is being developed at the Centro Universitario de los Lagos, under the guidance of Dr. Zuriel Cisneros Garcia and Dr. Jaime Rodriguez Zavala. To begin, this presentation briefly introduces metabolic disorders such as obesity and type 2 diabetes mellitus. Under normal physiological conditions, insulin binds to the insulin receptor, activating a signaling cascade that promotes the translocation of GLUT4 to the cell membrane and allows uptake. However, protein tyrosine phosphatase 1B, or PTP1B, negatively regulates this pathway by removing phosphate groups from the insulin receptor and other signaling proteins. Increased PTP1B activity can therefore contribute to impaired insulin signaling and insulin resistance. At the same time, mitochondrial dysfunction can increase the production of reactive oxygen species, or ROS, leading to oxidative stress. These two processes form the basis of the dual therapeutic strategy proposed in this research. The study evaluates C60-PORPHYRIN derivatives' two complementary functions, ROS scavenging and PTP1B modulation. Through molecular modeling, the aim is to investigate whether a single fullerene-based system can simultaneously contribute to reducing oxidative stress and modulating insulin signaling. So, how was this study carried out? First, the three-dimensional C60-PORPHYRIN structure was built in Gauss view and optimized using density functional theory at the B3LYP6311GDP level. Vibrational frequency analysis was then used to confirm that the optimized geometry corresponded to a local energy minimum. Next, the radical Fukui function, F0, was calculated to identify the most reactive carbon atom for hydroxyl radical addition. From this site, 4 possible configurations were evaluated: 166 bond, 156, and 2 para positions. Each proposed isomer was optimized and compared using its relative energy. The lowest energy structure was selected as the most stable configuration and used as the starting point. For the next hydroxylation step, the same procedure was repeated until the most stable derivative, containing 4 hydroxyl groups, was obtained. Next, the thermodynamic viability of the antioxidant process was evaluated. The reaction was modeled as a sequential hydroxylation process of the C60-PORPHYRIN derivative, using hydrogen peroxide as the radical source. In the first step, the system forms the dihydroxylated derivative, and in the second step, the tetrahydroxylated derivative is produced. For both reactions, the Gibbs free energy change, or delta G, was calculated to determine whether the hydroxylation process was thermodynamically favorable. This approach allowed the evaluation of the energetic feasibility of the proposed radical scavenging mechanism. Once the most stable hydroxylated derivatives were identified, the next step was to explore how they interact with PTP1B. Molecular docking was carried out with autodock vena, using the crystallographic structure 1NL9. For each derivative, 100 possible poses were generated, and the most favorable complexes were selected based on their binding energy and interaction pattern. These selected complexes were then taken into molecular dynamics simulations with GROMICS, using the Gromos 54A7 force field. After the NVT and NPT equilibration stages, each system was simulated for 100 nanoseconds to examine how stable the protein fullerene complexes remained and how their molecular behavior evolved over time. The optimized C60-PORPHYRIN structure is shown on the left and was used as the starting point for the thermodynamic analysis. The first hydroxylation step, leading to the dihydroxylated derivative, showed a Gibbs free energy change of -38.08 kilocalories per mole. The second step, forming the tetrahydroxylated derivative, showed an even more favorable value of minus 40.7 kilocalories per mole. These negative delta G values indicate that both hydroxylation steps are thermodynamically favorable. Overall, the results support the viability of the sequential hydroxylation process and the potential of the C60-PORPHYRIN system to participate in radical scavenging mechanisms. The docking analysis was first examined through the electrostatic potential map of the non-hydroxylated C60-PORPHYRIN derivative bound to PTP1B, the I. Active site region shows predominantly electropositive character, while the area surrounding the WPD loop is mainly neutral to electronegative, creating a complementary environment for ligand binding. All three C60-PORPHYRIN derivatives showed favorable docking energies. The non-hydroxylated system presented the most favorable value at minus 11.9 kilocalories per mole, followed by the dihydroxylated and tetrahydroxylatatives at minus 11.5 and minus 11.1, respectively. Overall, hydroxylation slightly reduced the predicted binding affinity, but did not substantially compromise the interaction with PTP1B. The non-hydroxylated C60-PORPHYRIN derivative showed the most favorable docking energy, at minus 11.9 kilocalories per mole. No hydrogen bonds were detected. Instead, the complex was mainly stabilized by hydrophobic and pi-pi interactions near the PD loop and alpha-3 helix. This suggests that the aromatic surface of the fullerene porphyrin system plays an important role in its strong interaction with PTP1B. After the addition of two hydroxyl groups, the binding energy slightly decreased to minus 11.5 kilocalories per mole. However, hydroxylation introduced two hydrogen bonds with Glu-186 and Ser-187, while maintaining the general binding orientation near the WPD loop. Reduction in binding affinity may result from a redistribution of interactions caused by the increased polarity of the derivative. With four hydroxyl groups, the binding energy decreased slightly further to minus 11.1 kilocalories per mole. Two hydrogen bonds with Glu-186 and Ser-187 were still maintained, together with hydrophobic interactions around the WPD loop and alpha-3 helix. Therefore, although hydroxylation reduces the predicted docking affinity, it introduces additional polar interactions without changing the overall binding region. The RMSD analysis showed that all PTP1B fullerene complexes reached and maintained structural equilibrium throughout the 100 nanosecond simulation, with no major conformational deviations. More importantly, the RMSF analysis revealed a clear reduction in the local flexibility of PTP when the fullerene derivatives were bound. Compared with the free protein, the ligand-bound systems showed markedly lower fluctuations, particularly in important regulatory regions such as the WPD loop and the alpha-3 helix. This reduction in molecular motion suggests that the fullerene derivatives stabilize and restrict the flexibility of PTP1B, which could interfere with the conformational movements required for its catalytic activity. Here, the molecular dynamics animations of the three protein ligand complexes are shown. Throughout the 100 nanosecond simulations, the motion of PTP1B in complex with each derivative reveals noticeable differences in protein dynamics. These differences are associated with the degree of hydroxylation on the fullerene cage, which modifies the interaction pattern between the ligand and the protein. Overall, the animations illustrate how hydroxylation can influence the conformational behavior and flexibility of the PTP1B fullerene complexes over time. In conclusion, hydroxylated C60-PORPHYRIN derivatives showed favorable thermodynamic behavior and strong predicted interactions with PTP1B. These results support their dual potential for radical scavenging and PTP1B modulation, providing a basis for future experimental studies. The authors acknowledge CADS and the Leo Atrox High-Performance Computing Facility at the University of Guadalajara for computational support. Veronica Rodriguez Celestino also acknowledges financial support from C60. Thank you very much for your attention. We hope you found this presentation informative and see you in the next video.